Good morning, everyone. Welcome to day 4. Today is a day that I think many of you have been really looking forward to. It is our sort of interactive-based, discussion-based, discussion-heavy day of the workshop. Today is really a day for synthesizing all of the conversations and discussions that we've had so far and thinking about the way forward, contextualizing everything that we've learned and heard and all the gaps that we've pointed out, and thinking about the paper, the workshop report. Today we have 3 modules. The first one is going to be our panel, leveraging the abiotic background. This is going to be an interactive discussion. We're going to think about applying the abiotic background to the potential of future life detection missions. And then after our break, we're going to have module 6, which is going to be the abiotic background in the context of life detection. This is going to be a really fun breakout. We're going to think about applying our knowledge in a mission context. We're going to have several activities where you will either work through an SDM or look at mission risks associated with mission concepts to ocean worlds. And then our final module of the day is going to be a wrap up and thinking about next steps and the way forward. And this is really where we're going to think about inputs into the workshop report. So we will break out and think about challenges, gaps, opportunities, and how to address those challenges and how to put all of that, capture all of that into a recommendations and synthesis and summary report. Okay, so with that, I'm going to introduce Tori Holer and take it away with our Module 5 panel.
All right. Yes, you can hear me. Thank you, Svet. And one thing that we really were hoping to do with today is sort of serve a synthesis function. So to take stock of what we've learned in the first couple of days and do the gap analysis and figure out as a community what are the important next steps for us to take. Another thing I was really hoping to come out of today's discussion is that there's a tendency, I think, at least in the life detection community, to think of the abiotic background as a challenge to be overcome. I would also really love for this group to think creatively and constructively about how we can conceive it in a different way. How can we use our understanding to advantage? And I think that will be a really important topic for us to explore today. So what I hope from this discussion is that it will be very interactive. If it goes well, I will speak very little. All of these folks will speak, all of you will speak. I would ask that you think of yourselves not only as people who ask questions, but people who participate in the discussion. If you have some relevant perspective to add, please speak up and please do. We have a few seed questions, but the hope is that we really get going. So to help explore this topic, we have assembled a panel of eminent scholars. They radiate wisdom. I wish that you could be here. It's like sitting next to a campfire of knowledge.
It's just gas.
As you will undoubtedly learn during the course of the discussion, Ram Krishnamurthy, Lori Barge, Heather Graham, and Andrew Steele. I feel compelled to call you Old Steely, even though there's no other Steely on the panel.
It works. It works. Classic. Yeah,
yes.
classic.
But objectively, this would make me Old Tory. So I'm on the fence about whether to do this. So that I have as little responsibility as possible in this process, I've asked each of our panelists to sort of introduce themselves, but also to as a starting point to offer their own perspectives on what they've heard this week, either as something they think is really important to repeat and resonate with, or something that has not yet emerged in the discussion that is important for each of us, important for us to consider as a community, putting out there as something that we need to consider. So Steely, since you have the mic, maybe would you be willing to introduce yourself and then offer your perspective?
Yeah, I'm Andrew Steele. I'm here at the Carnegie Institution for 2,000 years now. And I got involved in all of this back in the Allen Hills days. As a young microbiologist, I was brought into JSC to be the microbiologist to help with the discovery of life in Allen Hills 84001. Didn't go well. Had to leave after a wee while, and eventually ended up here. So I've been working on life detection in Martian materials since around about '98, and doing this, trying to kind of come up with ways of finding a microorganism they have no idea of the makeup of that microorganism against in any way, shape, or form, maybe against the background that you need to do initially. That was contamination because we found that the Allen Hills meteorite was full of penicillium, actually. Having cultured all of that, and everyone saying there was no life in it, and all of this huge research effort, it was kind of, wait a minute, we can't see the wood for the trees, so we need to come up with a better way of doing this. And that's what I've done for the last 20 odd years. And the abiotic background is, as these guys know, is very dear to my heart. Let's leave it there. So.
Hi, I'm Heather Graham. I'm from NASA Goddard Space Flight Center. I run the Agnostic Biosignatures Collective there. We're a laboratory that thinks about tools and techniques for thinking about generalized life detection. So not necessarily relying on biosignatures that we know and love from our own heritage as living organisms. I actually come to this field through paleoecology. So thanks for welcoming me. It's, as Andrea would probably defend, it's not as useless as you might think. But like Andrew, I find myself whiling a lot of time in the abiotic background because that's really one of the most effective ways for me to think about how we deconvolve the really complex signal that life might be when we don't have familiar signals to rely on. So that's what brought me into meteoritics, was trying to get more intimate with this thing we call the abiotic background.
Right. I'm Lori Barge. I'm a research scientist at NASA Jet Propulsion Laboratory. And I've kind of spent my whole career honestly studying these interactions with organics and minerals and the environment. And now, you know, working on missions and talking about life detection, it's just interesting because I've done so much work on, you know, looking at what minerals might precipitate, like when they have organics around and they make all these patterns, or when you have organics doing redox reactions or synthesizing other organics in the presence of minerals or other conditions. And so it's, when I think about the abiotic background, I think which one? Because there's so many depending on exactly what condition you're talking about. And so I think that's when I look at all the planets too, if there's anything we've learned from planetary science, it's you don't really know what you're going to find till you get out there with a spacecraft. And we've been surprised so many times. So it's pretty hard to predict what those conditions will be. And therefore it's hard to predict which abiotic background you'll be talking about anyway. And so I study origin of life as well, which is really a severe escalation of the abiotic chemistry, but by definition, it is still abiotic until you've actually started life. And so that's also something we have to consider when we're comparing to what a life detection might look like.
My name is Ram Krishnamurthy. I'm a professor of chemistry at Scripps Research Institute. You're going from the most involved to the least involved in this subject. So please pardon my ignorance. I'm a synthetic organic chemist with deep interest in trying to understand why life chose the type of molecules it did. So we make alternates to what life does and then compare the properties, what is gained, what is lost, and then try to understand why life is so special in terms of the molecules it selects. In terms of the prebiotic chemistry that I do, similar to what Laurie said, it's abiotic chemistry in the context of origins of life, which makes it prebiotic chemistry. So, that's how I see it and how I deal with it. So all of my research is prebiotically inspired, but not prebiotically realistic, which is probably true for 100% of the research in this field. People claim prebiotically plausible, so we just have to be careful with that. And so my take, I learned quite a lot from eminent scientists here and talks, and I was quite impressed with what all you do. You have such cool pictures that we can never compete with, the exoplanet community, and you guys take the cake when you want to put something on the cover of a magazine. We can't compete with that. So that.
Yeah, you could. So do you have more that you would like to say as sort of an open perspective on what you've heard that resonates, what you think needs to be said? Should we start there?
I kind of, yeah. I mean, just addressing one of Laurie's points, and I have a slide on this, but is the abiotic background changes per body that you're going to, because we don't know the reactions. You're quite correct, but life, you make two small assumptions. One, that life is carbon-based and that it doesn't use the entire alphabet, and whatever language is available via the abiotic background on whatever, from Titan to Enceladus to Europa to Ceres, to the bottom of our own ocean, whatever that language is, life will choose only a few letters. It will concentrate what it needs, and then it makes polymers, complex polymers from that. And so life in essence, when you break it down, is quite simple compared to the complexity of the abiotic reactions we can see. 10,000 and odd resonant masses in Murchison, 5,000, 6,000 on Mars in the Martian meteorites. We're seeing all of this plethora of chemistry, and they're different. Actually, I can show that. Might as well do it now.
You might need the mic, Steve.
So,
On the side.
so on this top left is FTIC RMS, and if you just look at this as the occurrence, you just get these dot diagrams like was shown a few times yesterday. But if you actually look at the concentration, and you bear in mind that for most abiotic reactions, the more complex you get, the less you'll have, like a Schultz-Flory distribution as being a good example of that. You see the Murchison meteorite. The Murchison meteorite at the top is 10,000 different. Down here is Ticint. Now we know from Ticint that above 600 degrees C, there's a refractory carbon component that we use to verify the Curiosity data on Mars. So there is a complete handshake between the in situ Martian data and what's in the Martian meteorite Ticint, which was a fall that we got really quickly after it fell and started analyzing it. So it was free, Mars sample return for free. It's a little bit of contamination in that meteorite, but not much. And then the top is one of my favorite things, a complex microbial fermentation consortia. And you can see that life is concentrating what it needs against these backgrounds. The abiotic background of Mars and the abiotic background of Murchison are completely different. So what we're looking at on Mars is not chondritic infor. It's a different type of reaction. Now, on Mars, that organic material in Ticint has a D to H of 3,000 per mil on average. And that's been done. I don't just do extraction-based stuff. The TEM at the top there, that's actually what organic material in Martian meteorites looks like. It's silica rich. It's amorphous. There's no real crystalline entity to that. And if you want to find life, you can. Terrestrial microbes love Martian meteorites, by the way. This is NWA 7034 and 3 little cyanobacterial cells. 3 little cells of cyanobacteria living inside this little crack, and that's a submicron Raman map of those cells. So we need both in situ, spatially resolved, as well as molecular based techniques that extract, and the baseline is different. You can see down in the bottom left-hand corner, Mars has an entire slew of aromatic carbons, the chlorine substituted, nitrogen substituted, oxygen substituted, magnesium substituted. No one saw that one coming, right? So, and sulfur substituted. And that's what we have to deal with. This is your background. So what would life do to that? That's the next question.
Thank you. Heather.
Yeah, so kind of riffing off of that, when Tori asked us if we could prepare a question to get this discussion going, I said, well, I'd like to bring in discussions about energy. And he said, well, I might lecture for an hour about that, which he made it sound like a threat, and that sounds delightful.
Talking about Tori, we're going.
Yeah.
That's why he kept his mic. No, what I want to add to this, and this is a great kickoff point, and the questions that people are challenging us with are great kickoff points for thinking about what are the physical measurements we would need to start to understand how we can model these chemistries wherever we go. Because as the point has been made, the abiotic background is different for each body. And it's also dynamic over time. But if we know if we have our organic observations and we understand the physical system, we can start to think about modeling how we think those things actually would have arose. Does it make sense in that environment, given the energy that's available and the material that it's there, that abiotic mechanisms are what is prevailing in that system? And I would like to remind everyone that while that sounds hard, and you're all thinking that's hard, it's not impossible, it's just PChem, so we can do it.
Yes, I think that is a great intro to what I was going to say too. It is PChem. And I think, so there's two things I think is important to take into account. One is that the abiotic background is different, not just for every planet or every environment, but it's really different in the granular sense, depending on the very specific conditions that are in that sample or in that microenvironment. And so coming from a background of origin of life, as I've always talked about this with Ram, like whenever you change the conditions of an experiment, say you change the pH, or you add this mineral instead of that mineral, or you have a different salt ion or what have you, you can then change the outcome of those reactions quite dramatically. And it's not to be thought of as I'm synthesizing a product, get a yield of a product. It's as a chemical network. And so you have precursors and then the products they make, and how that network expresses itself is going to be determined by the fine tuning of those environmental parameters. And so in the same environment, in the same world, you could have a variety of abiotic backgrounds simply by having, a mineral here that's different from the mineral here. And so it's very complicated, actually. And so I personally recommend that when we talk about what sorts of things are the background in different planets or samples, we should consider conditions rather than talking about environments, because environments is a type of thing that you can find on a planet, like a hydrothermal vent is an environment, but there's so many types of vents. And you could talk about, say, Prony Bay, where it's experiencing wet-dry cycling and it has carbonates and radiation. Or you could talk about a black smoker at axial seamount, where it's high temperature and sulfide rich. These are not really the same thing at all when we're talking about how they might promote chemical reactivity. So rather than say in a hydrothermal vent, we should say in an acidic condition promoted by a sulfide mineral or something like that. And that would help us, I think, connect better things that we talk about in specific samples with the types of environments we might look for. And I'll say more about that later, I think, when we get to our questions.
So you can see we already pre-planned, right? No, it's just flowing. So taking off what Lori said, as a prebiotic chemist, my question coming into this was, when Tori asked, what's the single question that you would have after hearing all these talks is, how can we prebiotic chemists or the experiments we do be part of your endeavor in terms of understanding the abiotic background? How does it help or where does it hinder? For example, yesterday you saw from the talk of Sarah that, the tholins in Titan, and therefore the type of chemical experiments that were done in Sagan's lab and others' lab to understand what type of tholins would be formed and whether there was even any correspondence based on what they found here when you actually made the observations, right? So the question for me is that when I look at your observations, for me, those are the most prebiotically realistic parameters that I can use to constrain my own work. When I see the slew of chemicals in a meteorite or the analysis that you provide, I say that's, prebiotic chemistry at its earliest or at that condition that can tell me what is realistic. But what we prebiotic chemists do is a very good trick. We take only the compounds that we want and then say it's prebiotically plausible and then get it through the editors and reviewers and get a good paper and then the headlines. So sometimes I feel that we do this, for the sake of the students to get a good publication so that they could get a good job and move on and things like that. But at the same time, I wonder in your bigger picture of when you are struggling to ask these questions, the abiotic background versus the biosignatures, how can we help? Or, what is the message that you would want from us, the type of experiments that would help us move not only your field, but also in the long run help us in prebiotic reality?
I think, what we're doing is actually helping you from the get-go, because if we don't find life, if we don't find something that pops up above this background, it's informing abiotic chemistry. And at this point, we're taking the experimentation out the test tube in the lab and into real environments and studying those. And that gives us an insight into how life started here and helps us to help you constrain those conditions so that instead of choosing the ones you want, you can choose ones in a planetary relevant environment and then start to really interrogate that. And so this interplay is backwards and forwards. It's iterative over time, dependent on the observations made. But for instance, some of the organic chemistry we've seen in the Martian meteor, I'd love to chat to you about how that could change or be altered or become something that you would start to imagine could have helped with life on Earth. But, you know, nothing's popped up so far on Mars that actually makes us think that there's life there. And life tends to buffer the environment as well, doesn't it? It tends to produce what it needs to control that environment for itself. And so, whereas abiotic chemistry is a slave to the environment, as Laurie was saying. So that again, in of itself, is a good way to look at how you can use this background, to basically do life detection.
Great, thank you.
So we're going to help you.
You're welcome.
It's going to be a merry dance.
Group hug, come on, bring it in.
Bring it in, guys.
So one of the questions that for me motivated this workshop was to often in life detection, we think about, you know, building evidence and an expectation that grows our confidence that what we're seeing is evidence of life. And so I wanted to sort of flip the question and ask, is there a way that we can build confidence that what we're seeing is not life? Do we have a sufficient expectation of what abiotic looks like that we can make observations that help to support that hypothesis? And Barb gave us a beautiful example of this yesterday, right? That this screening process as to whether or not you release return samples for further analysis is based first on an assessment that you have confidence that what you've seen is abiotic based on the patterns that you're looking at. And we've also heard articulated really beautifully here that the abiotic background is not one thing. You expect that it's going to be different everywhere you look. And so what I want to ask you is how we reconcile these two things, especially in light of the fact that you know, when we go looking elsewhere, we'll have a lot of uncertainty about what the environment is. We won't know beforehand necessarily which environmental factors are the most meaningful. So I want to ask you, is there a subset of things where we think we do have a sufficient expectation of what might look abiotic, that there are some commonalities in how we can make that assessment?
Oh no, I'm holding the
Yeah,
cube.
you're
I feel
it.
like I have to answer this. Let me think about that. I think one thing I would say is, this is, you know, since I already brought up it's not even 9 and I brought up PCAM, so I'll just keep digging a hole, which is doing that dreadful thing of targetless analysis. Really try to characterize your system. This is the peril of designing a mission for life detection, is you're making some assumptions about what you think you're gonna see. And when you make those assumptions, you're probably pre-selecting against certain things that could become really important farther down the line as part of your analysis when you are trying to compare some weak signal to a strong background, and you may or may not have all the information you need. This is why, you know, what I was trying to encourage everyone to think about is what are those contextual measurements that you need? And sometimes, and a lot of times, they're not going to be chemical, but what are all those contextual measurements that you're going to need? What are the physicochemical things that would help inform your interpretation of any measurement or data set that you develop? I think one of the best things we can think about as far as the complexity of all this nonsense that's over my head is that it does give us a starting point for what we think starting materials are, as far as just reduced chemistry. And this is important because when we're looking at reduced carbon on Earth, we're just looking at stuff we've inherited from organisms before us. So there's already a bunch of pre-selection that's already happened on that pool. And that's why we need to think more broadly about something that hasn't already gone through 4.5 billion years of biology.
I think, you know, Heather brings up a great point about having a holistic way of analyzing the samples, because a lot of our techniques are actually built for oil industry and geochemical point, whereas so they're all automatically biased towards a terrestrial understanding, and having techniques that you can have a more agnostic analysis to understand the slew of stuff, but is good. But the understanding is that this is a process as well. It's like one measurement is not going to get you a life detection. One measurement is not going to set your abiotic background. And it's both extraction and in situ understanding. There's things like morphology, the mineralogy, the context. All of those play a part and a role into understanding what that prebiotic background is, because it doesn't necessarily have to be organic. It can be morphological or it can be mineralogical. Yeah. So all of that plays into it together and invokes a process of discovery that also includes Ram's experiments and Laurie's experiments, and trying to ratify it. When you're on a mission, you get an observation and then you go to the lab and you try and reproduce that observation to understand it better. So in that, there's a process that needs input. So.
Yes, I would agree with that. And I think it's important to remember, so in a mission context, even if you could have perfect organic detection, which maybe we won't, or if you could have a perfect sample analysis from wherever, it's even if we have all the data we want, it's not necessarily going to be possible to interpret that right away. And so I think, and I know this isn't always the news you want to hear, but if you have a perfect organic detection of a sample, say from Mars or Enceladus, it's going to require some lengthy comparisons with lab work and tests to find out how different parameters affect those organics, let's say, in that sample. And so, for example, you know, we see it's all over the experimental literature. You can see how different organic chemistry is affected by, say, changing which mineral is present in the solution. Or even if you switch which precursor it is, if it's the same type of molecule, but that's a different version of that, you get a different result. And so we cannot say that just because we have detected some organic sweet in a sample that we can tell whether it's abiotic or prebiotic or biotic until we have had a real good complement of lab work to compare. And this will take time. And some of this can be done alongside a mission while it's already going in advance even. Some of it is going to have to be done maybe after we get the detection. And there's just not going to be a quick answer right away, I'm afraid, for a lot of these scenarios.
Oh.
So as an organic chemist, when I read, the headlines, building blocks of life found, in meteors or things like that. One of the questions that I always ask is, how do you know they are the building blocks of life, when life does not exist there? Right? We are putting our idea of life on Earth on them, on that sample, and we get excited when you say, we see a molecule like uracil, purines found or pyrimidines found. But the problem is this, let me tell you, because this was very hard on me because I had to change my views. I was brought up like, when you get into the field, you get under your mentor and then you kind of follow the pathway, and then you have to kind of go your own way. And those are the painful points when you have to kind of separate yourself. And this was with respect to RNA. And so the problem was, I was explaining to Tony that we get excited by seeing uracil or adenine in the meteorite, right? Because it's a building block of life for us in biology on Earth. But the problem is, nature did not select that uracil based on just that being uracil. It selects only uracil in the context of an RNA as an oligomer, where it expresses a base pairing property in the presence of a complementary base pairing partner. Until then, uracil has no meaning. So if function was the selection for this particular molecule, we can't get excited about the molecule where that expression is, the function is not expressed as a single base. So when I see people saying, oh, building blocks, amino acids found, again, the same kind of things come about. So My question here, with respect to all of this is a little different because I struggle with this every day, how to justify the type of chemistry that I do versus the type of observations that you make and the types of claims that are coming out, because that is what is motivating this whole, the question is, how do you justify $1 billion or $1 trillion investment? And the question is, are we alone? Right? I'm sorry? I'm trying to motivate the funders.
Remember, Morgan, this is the optimistic day
Yes.
of the workshop, so.
He told us to be optimistic,
Yeah.
so that's the question that we ask. And the question is, how are we boxing ourselves by that question? And are we kind of, coming back to that question and forcing that question on these kind of analysis in such a way that we are forced to give an interpretation that we ourselves would not be comfortable with?
What was this?
The other one, so it can go to the audience.
Yes. So I have many, many questions, but I don't want to be greedy about it. And so I do want to open it up to all of you to participate or to ask.
Kristen Johnson-Finn, RPI. So something I was kind of thinking as you're discussing that we're dancing around this point that what we're doing, and somebody who likes organic compounds, I'm quite biased as well, but like organic compounds in of themselves are not a good metric alone. And like Lori's doing experiments where it's morphology, and there's other people doing experiments with different kinds of factors. So what sounds like to me is that we want actually is a list of combinatorial factors that give us information about the context of the system and how that impacts possible life detection. And so I was trying to think of like a real forced analogy. And I was like, if you walk into the breakfast nook in the morning and you smell coffee, you don't know if that's one cup of coffee or coffee for everybody. And you're assuming that you already know that there is that coffee is something that you can smell, right? But if you just smell that mix of compounds and you don't see cups and you don't see the carafes and all this other stuff, then you don't actually know what you have. You just know that there is something you're detecting. And so there's a contextual component that I think that I definitely don't yet have a handle on, but I think that the group of people that's here, we could actually come up with a good list for like when you have an actual rover or a probe or whatever, there's the context of your limitations of analysis, but then how do you choose the best way to get the best of both worlds, and how do you do experiments to help give that informational database to give us that information? So, thank you.
I would just add to that, two things that I 100% agree with you that from paleobiology, most life detection has not involved organics. That's not how we've been finding life in the geologic record. And I would like to add to that, that when we talk about this contextual thing, geology and time itself becomes part of that context that's necessary. Because we want to make sure that if we're thinking this is contemporary, is it really contemporary?
I sort of agree. I mean, life detection is reliant on context. And that's what I was saying earlier. The list of, there's 27 different techniques that we applied to SIP, starting with just simple, what kind of rock is it? Starting with a camera and working all the way through to TEM and sticks them in from centimeter to nanometer scales. And you're going to have to do all of that. There's no two ways about it. And it might be that it's an organic poor fossiliferous tape that you get, that you see gunflint type microfossils. I would argue that Strelly and all of those aren't microfossils personally, but I think most of the paleobiology is seeing dragons in clouds. It's so we have to guard against that as well. Certainly in those really old samples, but that's a talk for another day.
Chris German from Woods Hole. Ram, I really appreciated your point about the prebiotic chemistry is kind of very selective of you what's plausibly prebiotic. Is there any feasible, this is completely naive, but is there any way you could go to the other end of a life process and say, okay, well then, picking up on what Steely was saying, is like life selects from a few letters from whatever alphabet is available. But presumably it does it in such a way that the metabolic waste products might have some kind of characteristic to it. So when Heather was talking about, everything on life, the gunkiest organic stuff ultimately was always there because of it's a biologically processed thing. Is there a way one could try and look at what are the resultant products, and if those are different in the same way that life fractionates compared to what's available as raw material in abiotic signatures, could one then theoretically think agnostically of there's always going to be a package of, maybe things are going to break down from large molecules to certain patterns of small molecules that might be metabolic waste, that one could work out how one idealized would look for, and then exploration is basically seeing something different from what you were expecting, right? That's discovery. So is there a way you could then turn around and say, okay, you can go and look at something where you want to say it's abiotic, and can you sustain that because there's nothing remarkable, there's nothing weird in the patterns there? So could you do like the inverse for that?
So what I was going to say, I think this relates to the previous one too, is it would require lab work and it would require a lot of testing for those specific scenarios. It's possible that a biological system could generate certain waste products, but then you would have to look, like imagining you found some suite of organics that you think are the waste products. You'd have to look at carefully whether those could be produced by any other abiotic means. And then also it's not even just the production of things through, say, prebiotic processes, it's their processing they go through geologically, as you were talking about. So even something like, which of these molecules are more likely to be sequestered by the mineral versus lost to the aqueous solution, or which one is more likely to be reactant with a volatile that's present, like say ammonia, and then, or something like this. And so all of that also has to be tested. And in my experience with these types of experiments, it's not predictable. So it's not a good idea to say, well, based on this like similar-ish reaction, I expect this is the case. You have to actually do the experiment because the results are often very different. The good news is there's a lot of other literature in other fields that have done a lot of this. And I don't think as in planetary and astrobiology, we are often aware of this other literature. So it's things like environmental science has often done a lot of work about mineral absorption of organics and transformations, production of certain compounds we're always interested in, like gases of nitrogen and carbon. And just there's a lot of other fields that we don't interact with that have some of these answers already. So that's the good news. And I think having more connections with people who do these types of very well-funded work in comparison to some of our things, and have a lot of data about the different outcomes of different organic reactions in environmental systems that are relevant to the planets. So that is one thing I would suggest.
So just wanted to add, because Steely was referring to this, what, and this is a question that I would like to ask to the community is, what would be a definitive biosignature for you guys, when you say, aha, that would be something that would be unquestionable. And one of the things that I think I heard was polymers. If you found polymers, you would consider or would tend towards that?
What?
Yeah.
Yeah.
We got lots of PAs.
Yeah, yeah.
Yeah, a combination.
It doesn't exist.
Okay. So because, the polymers also can be produced in an abiotic fashion. And so, for example, Moran referred to it briefly in our talk, these kind of mixture of alpha hydroxy acid and amino acids, which you find in meteorites, can give rise to depsipeptides, which eventually will go to peptides in an abiotic wet-dry cycle. So one has to be kind of careful about, whether you would use one criteria. As I said, you have driven the point home more than enough, saying that it's contextual and it's not a single observation point. So the question is, if this community can kind of settle on what would be a biosignature, what would be the type of confidence levels that would be, and if that's not attainable, then we have to kind of wallow in this, abiotic versus biotic background. Yes.
So I think my first instinct is that's never going to happen.
Okay.
Except that if we can agree on a couple of really key features that life performs, like, a lot of what we're talking about as being cool signals is energy put into a system so that it differs from the abiotic background. If we could accept that, then we could probably get somewhere.
Yeah, I think, you know, the realm of proteins and nucleic acids, the complexity of those polymeric systems is huge in life, but when life dies, it decays. In meteorites, we've only found what, diglycine. There's been some.
That's the most.
Yeah, and so if you do see a polymer, again, it's a process, right? You start to kind of really zone in on that polymer, and if it's just a single repeating unit of something, it's an interesting thing, but it's difficult to understand how that would be life. And so, but then it comes back to lab work and looking at it and really kind of studying that for what it is, and try to understand how that could relate to what we would describe as a life process. And certainly in terms of the sample safety assessment group, whether or not that is then a threat to an Earth biosphere or life here. And that was the thinking along those lines. But also, like, things that we take for granted for life, say racemization. You know, you can date the age of shells from the extent where that life signal is going away over time. So without the morphology of it being a shell, without the mineralogy of it being a shell, the amino acids are going to tell you there's no life.
Yeah.
So it has to be a holistic approach to all of this.
Thanks. These are wonderful conversations, and I violently agree with everything everybody said about the complexity and the way forward. But I was caught by Ram's word about wallowing, you know, how do we ensure, and it is true, anytime you're sort of at a moment of perhaps a paradigm change, and I think we may be there as a community. What can help is to focus on the small victories. So what I see with this group is around the communication, right? If we can really make that change, that as Tori opened, right? The idea that abiotic baseline is not this thing over here, that the two are indivisible. The search for life is abiotic baseline. We recognize this in Chem 101. No signal makes any sense if we don't know the baseline. So we all agree, but this hasn't been communicated. So that's a huge victory in terms of the use of abiotic baseline, just to begin to, or continue to articulate it in that sense. And then the second point I want to make is related to that, which is, and I think the SSAP exercise was an example of this. This idea can be powerful even before we've done everything we need to do. Because what it provides us with is an ability to look at the knowledge at any point that we've got it. It allows us to do things like Lori's suggesting, to say, okay, in these systems, we may have some idea of an abiotic baseline, but in these, we've got to do this much more work. So it helps us define research paths forward. It helps us define where to put our energy. Somebody else said, well, and this is absolutely true, it's not just all about abiotic chemistry. There's abiotic baseline principles in geomorphology, in inorganics, in mineralogy. So again, it helps us to identify where we need to go. But similarly, it helps us solve problems right now. And that's the main thing I want to say. What the SSAP showed us is that even while recognizing that we do not know anywhere near as much about the abiotic baseline or biosignatures as we want to, it still allows us a decision-making factor if we are faced with a problem right now. Because we can take a look at the sum total of our knowledge and say, all right, that analysis or that approach is this far away from perfection, or we've got something that's a working testable model. And so it allows us to prioritize what we want to do and where we want to go, both in terms of immediate actions and in terms of potential long-term research. And so the key thing I want to emphasize is that it's powerful even in the early stages. Perfection does not have to be the enemy of actually using this in a very powerful way for certain things right now.
That's a great point. Thank you.
Just quickly to add to that. Look, we don't know what an organism on another planet would evolve to be like. We can sit here and we can write science fiction books until the cows come home. But one thing that we can do is look at chemistry. we look at the chemistry of systems to begin to try and understand what that chemistry is and let it open our eyes to how that chemistry was selected for life here, as an example, and then extrapolate that out. And so it gives us that baseline just to give Barbara's comments some context.
Go ahead, Jen.
Hey,
Okay.
Jen.
So, mission hat on. I understand what you're saying as a biotic background on what's presented here and what you've discussed, which I think is great. I appreciate having that reference point. I think that it helps us conceptualize and kind of come together in our own minds, which are all different because we come from different backgrounds, on what the deviations from a biotic background might be and help us explore that. But if we're going to another planet or planetary body and we don't know the conditions, we don't know what the abiotic background is there, and we don't know if there's life there or if it's prebiotic chemistry, in all practicality, what is the abiotic background then? How does that help us go forward? Because we have so many unknowns going into a mission scenario that, we're going to get what we get. We're not going to get the abiotic background or the life. We're going to get whatever is there, and we get to interpret it. So
From that process.
I can't
Well,
tell
well, we
if
start
this is
from-
more question or assent or.
do
what's
we
happening.
do we assume
Just very
something?
eager people.
Or, do you see what I'm saying? Like, I'm trying to grasp, how do we use abiotic background in our,
You start from the point of view that everything's abiotic until you prove it definitively against that background, whether something cannot be explained by abiosis.
so it's not Murchison meteorite.
No, the abiotic
It's-
background is something that changes constantly.
Oh, yes, it does.
Yeah, but that's what I said earlier. That's why I showed Murchison and Mars. It's
But then
different.
how are you ever going to tell the difference between the abiotic background and something that is approaching life or is life?
And that's what I said earlier about it doesn't use all of that alphabet that it's available to it. So on Europa and Enceladus, as I said earlier, we have made measurements there and we have decided that most of that is abiotic, but it's interesting enough to go back and look, right? So we have those measurements and it's guiding us. So if you have ACT and G, you might have LMNOP. It doesn't matter, but life is not going to use or be a component of all the components in that system.
Yeah.
So it's going to concentrate that up in a certain way, like
So
in the
I,
top right.
if I understand what you're saying,
Yeah.
is we're not going to these other places to test for an abiotic background.
No.
We're looking
It's
for
exploration.
something that and that abiotic background is something we conceptualize based,
Characterizing the chemical phase space.
it's something we conceptualize. It's not something we target for analysis with untargeted analysis, really.
Perhaps we should.
But I think that that's a really important distinction that we need to communicate to the community.
Well, and we are in the process of having like a box. So it just so happens, I think, we're concurrently in planetary science, having all these nice explorations of the planets and their samples at the same time as the community is understanding what life really even is. And so we're still working out that, what is the difference between life and non-life? Prebiotic chemistry is always at this question, like, I get asked a lot, when do you know when you've succeeded as an origin of life chemist? How do you know? And I mean, how do you know? Because it started abiotic. So like, it's the same question we always have in the planetary world. So we're kind of in parallel here, trying to understand what it means to be life in the first place, at the same time as we discover things and try to analyze them. And I think it's going to be an ongoing feedback of iteration here that's going to just take a while. I wish we already had the definition when we sent the missions, but unfortunately, we're still working on it.
So
Yeah,
Morgan and
Morgan
Shannon.
Cable JPL, I totally agree. And I think this is sort of where, by our own fault or not, the community is kind of we've sort of shot ourselves in the foot by having such a high bar for life detection, right? It's a binary thing. Is life there or not? And now we're holding future missions to this requirement. Are you going to look for life or not? Versus Are we just going to go and look for something interesting? Are we going to characterize the physical chemical conditions, the chemistry, the organics that could be there and what their evolution is? And so we've sort of backed ourselves into a corner now. And I think part of what we can do in terms of informing the community is to maybe find creative ways to walk ourselves back from that of, the success criterion for this next mission may not necessarily need to be, did we find life or not? It's, did we find something interesting? Yes, and so what comes next? Because this whole search for life, we all recognize it's a process, right? The next mission that's going to go is going to find something interesting, and the one that follows on that is going to build on that and have more specifically tailored instruments now that have been informed by those constraints on salinity, on composition, on temperature, so that we will have more diagnostic and pinpointed tests next time. But I think there's something to be said about the storytelling of how we communicate that within our own community and to the rest of the world, that this is not just, unless we go to a world that is so inhabited that it's like Earth, and then you just snap a photo and it's obvious, like there it is, right? So I think that's something that's on each and every one of us as we communicate to be more, I don't know what the right word is, but to be ambassadors that are open and honest about what our expectations should be.
I wanted
So-
to just add to that I think we should do a little bit more of a better job communicating the fact that we barely know how planets work without living systems overprinting nearly everything. Because that's what we have on this planet. We don't know what a planet without life really looks like.
Yeah, but if you tell a politician that, now you don't have funding anymore.
Right. But the thing is about life detection, I mean, NASA's got two main questions for NASA. Are we alone? And how did we get here? Right? And these, if you start at this point and go and look, you're doing both. If you don't find life, it's not a failure. You're finding abiotic chemistry and how we get here. So you're answering both questions. It's a twofer right from the get-go. And if we communicate that, well, depending on the current thinking in political thinking, it should be a win-win situation for everyone. And that iterative process means it's going to be over time that you actually solidify those thoughts down. And based on your observations, don't second guess what you're looking for. Don't say, this is the baseline I am looking for. Go measure it first. Then start to do the interpretation of that. So the abiotic baseline relies on agnostic measurements and agnostic analysis initially to define as you go as part of that.
No more.
Well,
So let's go to Shannon, then I want to give some time to online questions.
We have two other questions out here too.
So, when Tori and Svet and Alfonso asked me to speak about Orbulander, I was like, oh, so who's going to talk about Titan or Dragonfly, right? And I really pushed for that because we didn't use, when we were sitting in a room creating our STM, we didn't use the framework of an abiotic baseline, but that's really what we're going at Titan, because we have such a low expectation of actually finding little microbes that are doing something at 90 Kelvin on the surface. But we know that there's interesting organic chemistry, and so we tried to create a suite of instruments that are getting at the environmental context, that getting at the chemical, diversity, to try and establish what is the chemistry that's possible in this environment. And we also happen to be carrying an instrumentation suite makes it possible to do like a search for chirality. And so if there was a really obvious enantiomeric excess, maybe that's a biosignature. So from the beginning, we have been trying to tell this story of, we're going to do these chemistry measurements, we're going to do, classify where they are in the environment. And the story that we've tried to tell then is that it's like a prebiotic exploration. And I know that's not a perfect term, but I think it's a really evocative and useful way to communicate like beyond this community, using that word of prebiotic. However, we all the time get classified as a search for life mission by people off the street, journalists. I've seen people at headquarters think the same thing, but we're not. Like, it's really hard to walk that line. So I would be very curious to hear, not necessarily as part of this discussion, but
before we all leave, what people are taking away from our messaging and like what you think, if we're doing that, defining the abiotic baseline and allowing the search for life to happen, if possible, in what we're trying to do. I don't know if that was a clear question, but like, I feel like Dragonfly is an opportunity to test these things because because Titan is so unlikely to have life as we know it at the surface. So if there are ways that we can do that better, we want to know.
So this is actually something we talk about a lot in Origin of Life. And I mean, I need more marketing, but searching for prebiotic chemistry is very exciting. And, you know, as Origin of Life scientists, we're very envious of the geologists and planetary people because you have so many examples of environments you can go to. There's life everywhere on Earth. You can find it all. Whereas we are stuck in the lab. And so it's like, okay, we'd love to see this somewhere, anywhere. You can't find it on Earth. You can't find it like so far on the other planets. You can't really see anything. And so we kind of just get to be in a test tube all the time. So if there could be a planet or a moon where you could witness any of this in the field, it would be just transformative in a way that, you know, we can never really have. And so this is really exciting to us. Marketing it to the public, I don't know.
Well, it's twofold, right? Like
That would be great.
I want to make sure that the marketing to us as well is correct because that's who
So-
evaluates
So
proposals.
Shannon, can you use this line? We choose to go to Titan because it is there.
You're hired.
I'm just going to reiterate that and to say, look, this is what exploration is of Titan, carbon nitrogen chemistry. That, I mean, that is, you know, we still don't really know how HCN chemistry could help. And, you know, there's a lot of Origin of Life scenarios that talk about HCN chemistry. And so looking at that, that's for me, that's the bit that you if you're not looking for life, you're looking for how that chemistry would potentially form in that environment. And you said it yourself when you showed that, what was it, Sean Donegal Goldman's pale orange dot, right? And the similarities with potentially a very early Earth. So those are the ones that if you're not looking for life, you're looking for how to inform how life could have started. And I think that that's the best message.
We've got some online questions. Elizabeth, go ahead.
Hey there. This is Elizabeth Spears from Woods Hole Oceanographic Institute. We've had a really lovely discussion today, kind of talking about so far about like, how do you distinguish the abiotic from the biotic, and you know, would a biosignature actually be? But I think one thing that we can maybe discuss a little bit more that would be kind of a nice pivot, or not pivot, but it would be actually looking at the abiotic and not just how do we differentiate it from like a biotic signal, but how can we actually use it as a tool? How do we actually leverage that abiotic signature? What kind of processes or reaction pathways that are abiotic in nature could we use as tools to help us in the search for life? Because, you know, it's this, obviously we've heard today, it's this really complicated problem to have this dichotomy of abiotic versus biotic, but I think also we can also think about how the abiotic can help us, even if it's not telling us necessarily what the biology is. What concentration processes could it be doing for us? How could it be moving specific compounds or not moving other compounds? And I think that's one direction we could take to kind of move this discussion in a way of looking at the abiotic as a tool rather than as like this opposition to biology. That's more of a statement than a question, but sorry.
No, that's fine. You know, often I have wondered, you know, for example, when we look at all the distribution of molecules in the samples that you bring back or the samples that you analyze, I always look for what is not there from biology. You know, for example, one thing that really stands out for me is the absence of nucleosides or nucleotides. You never find them, you know, that they are the basic building blocks of RNA and DNA, but you never find them. You find the nucleobases. There are kinds of sugars, which I have my serious doubts about, but you know, because sugars are not very stable and they will not survive those kind of environments. So the question is, why are there not nucleosides, but the nucleobases? You know, can that give us a clue? You know, feeding back into the type of, you know, the question that you asked, what are the abiotic process? Is it a process of destruction that we see, you know, and the bases are quite stable and they are left behind? Is that, you know, but there is no way of knowing the history because no information is embedded, as Steely pointed out, in that single amino acid about its history without placing it in a context. So why are we not seeing nucleosides and nucleotides in the, we see plenty of phosphates, we see plenty of sugar degradation products, like, people have seen some, Cooper has, seen some sugar-like acids, sugar acids, for example, but we don't see them. So why are we not seeing, for example, the nucleosides, which are the building blocks of RNA and DNA? So that would be something for you guys to think about in that context, maybe.
Ali Fox online has a question that's a bit of a pivot. So let me see if is your audio working?
Can you hear me?
Yes.
Yes.
Woohoo! Yeah, Ali Fox, NASA Glenn. This is kind of a pivot, but I think it relates to a lot of what we were talking about earlier. Given how hard it's going to be to have an authentic life detection where we feel confident that it's definitely life, how does this panel feel about life detection experiments similar to like the Viking biologic experiment?
No,
I
no
think,
strong feelings here.
So you're looking at more at metabolism. And so what you're looking at is change over time based on some baseline process, right? Ali, is that correct?
Yes, yeah, I'm thinking something where you could maybe watch it in real time. Yeah.
Yeah, I mean, time can factor in. Life does tend to change things over time. Depends on how you design those experiments, I would say. I mean, we know the problems with the Viking experiments. And on Earth, we've got less than 2% of the microbes out there that are culturable. So that's getting better a little bit, but the genetic diversity versus what you can culture and grow and watch over time. Orbital measurements and orbital missions have shown that it's safe for Mars or for some of these moons, that there is change over time, but how that relates to life is yeah, that's not really been hammered down a bit. We don't see swathes of green suddenly appearing in certain areas of Mars and then going away during the winter or stuff like that. Doing a reaction process or a reaction profile of that would really depend on doing precursor measurements, in my mind, to understand what you're trying to put into that system. Is it a seawater-based thing? Is it a media-based thing? If you're going to grow something or look to see how that life would take the atmosphere and change it over time against an abiotic background or things like perchlorate, it's very difficult. I think it's doable once you've done the preliminary understanding of those environments. Say if we look at Enceladus and we went there and we got a whole slew of things and knew the ocean chemistry and things like that, then maybe you could look at an experiment to kind of chart that over time, or a C14 release experiment or something more targeted. But at the moment, I would say that you're looking at morphology or organic inventory or other processes initially, but certainly time-based measurements could be key in understanding what life is, especially if you've pre-characterized something as interesting.
But before we go to a next question, can I just ask for the reality check for anyone who has a schedule in front of them? How much time do we have left? I know it seems like something I should know as the moderator, but if we're being honest, like I don't even know the code to my own lab, so.
What even is time, man?
So we have 5 minutes left, so this is going to be the last question.
Oh, I would like to come back on the first comment that has been done and build off on what everyone does. So from a practical question, when you go back to the lab or on your computer, what will be the first step you would like to do? Because we all try to do a list. We struggle to do a list because, like Laurie said, every environment, on every location, microscale location of your synthetic or natural analogs give different conditions and so provide different possibilities to organic chemistry to evolve, or even for microbes on Earth to adapt. So what will be the restricted list or restricted condition you will base of for spaceflight mission where you don't know what will be these conditions, or for return samples where we would have, with the curation center, have some preliminary result on the context to build up like the analysis or this stuff, because like I will maybe not ask this question because in biology we struggle, but if we find viruses or proto compartmentalization where the distribution of organics and the selectivity is present in this compartment, but we still say that it's not life, how should we say that we have a abiotic background or prebiotic background?
Well, you guys can speak to the sample analysis, but I would say an important part of this is going to be having a robust repository of information about what happens to these types of organics under different conditions, and that's a lab work project. And I would say that if we get more organized, and actually, we were talking about this yesterday about how things are reported, say from the origin of life or from the abiotic chemistry studies, like the experimental work that we do, it is really important to report things in terms of conditions. So for example, I could say those vesicles form under, well, you know what, I don't study vesicles. So let's say this protometabolic system forms better at an alkaline pH than it does in an acidic pH, specifically when you have a high amount of reduced iron. Like that's the sort of information we can use, because then, you know, from samples and missions, we can say, okay, reduced iron, and now you worry about where that's found on planets or whether it's in a sample or not. As opposed to a type of reporting where I could say, this protometabolic reaction is favored in a brine on land, when what I really meant to say was high carbonate favors the reaction. So things like that are super important. And we discussed this in terms of, say, high phosphate being interesting for certain reactions. But if you just say high phosphate, it's very nice because then you can go to Enceladus and say that could be here. Or you could go to a carbonate lake on Mars or early Mars or Earth and say that could be here. But not restricting the type of environment, but just stating the condition. This sort of thing, like as Laura was talking about for a database, could be very useful. And I would love, it would also save us so much work in the origin of life lab arena to be able to look up things like what happens if I add ammonium to that reaction that I already did. I don't have to do it now if somebody else did it, as long as it's reported in the right way.
Yeah, because we are multiple people to do databases, but there are so many conditions that lead to organic complexification that what will be the restriction? How could we constrain the list, because the list is extended, like it's infinite?
I think the way we look at it at the moment, you know, as far as ocean worlds go, is that
You need to learn to hug them.
I know. Is that yeah, the conditions can vary quite a bit, right? But the understanding of it is a process. It's not a list of things. So when you make your measurements, you start there as to defining what that abiotic background is. And you bring up a point about viruses and prions, which as a microbiologist myself are close to my heart. You know, you have repeating subunits, and those are polymers, and they're going to be interesting to look at. I'm going to want to go looking for a host, but still, who knows how life started here? It could have started through proto-life constructs like viruses or prions. We don't know. And it goes back to another microbiologist comment earlier. If you pop an E. coli very gently, its constituents are there, but it's dead, right? So when do we declare life? And that might come back to Ali's suggestion earlier, that it's life when it actually still does something and doesn't just lie there as a constituent with all its constituents around it, you know, mort. You know, so it's a process is the most important thing to remember.
So
It's a process that we go through at each point, making measurements, understanding those measurements, discussing them on panels like future panels like this, which, well, I won't be part of, you guys will. So, and just remember that make the measurement, and then let's discuss it, and then make the next measurement and discuss it, next measurement, and so go through that process.
So based on the last remark, we should do online, like in real-time analysis to see the changes, because life is a changes procedure.
Aaron, is this on the same thread, or is it a new question?
It's a slightly more optimistic point.
In the spirit of the day, then, Aaron.
So I guess I just want to point out that, I mean, we have one set of molecules that we think we like, right? I mean, we like the amino acids, not just because it's a biosignature, but because we think we know the baseline. We think the baseline is racemic, and they're simple enough that we think they're going to be there, right? So, you know, that's and I know there's obviously limits to that. It's not, but so it's sort of like, I think the hope would be that, you know, that's coming from a biology point of view. And to me, the hope is maybe there are some commonalities among abiotic processes that we could start to say, and yeah, these are some of the kind of course things we should be looking for from the other point of view as well. But, you know, yes, there's a wide variety of possibilities, but we have some footholds that I think we should work from.
Yeah.
That really comes down to the fact that we're here together, and deliberately is the life detection and the prebiotic chemistry. You know, are we alone? Where did it come from? And that interplay between us and S is the most important part of all of this, because we're not separate fields. We're all in this together, and it's a realization that we can help each other.
Yeah.
Right?
Group hug. So
Solidarity, brothers and sisters.
So group hug, but I also noticed our panelists starting to kick one another down at the end. So it might be time to just gently walk away. What I do want to say, I know that you still have questions, and that's a good thing, because this is meant to be the start of a day-long discussion. So we have several more permutations of ways to ask and try to address some of these same questions. So please join me in thanking our panelists for this morning.